Hello! Welcome back to our class in Chemistry 102B Laboratory. Last time, I gave you a formative exam on the common apparatus which are to be used in Analytical Chemistry Laboratory. So today, we are going to check your work, whether you have identified them and gave them their functions. So let's start with number 1. The first apparatus there is what we call the burette clamp. This burette clamp is used to hold the burettes, especially when titration process is to be done. The second is the burette. Now, this apparatus is used for titration purposes, and it dispenses very small amount of liquids or solutions. The third is the pipette. This is also an apparatus which is commonly used in analytical chemistry laboratory, used to dispense very small amount of liquid. Number 4 is the reagent bottle, and this is a container for reagents or some chemicals. Number 5 is the separatory funnel, and this is used to separate two or more substances which are immiscible. Like for example, when you have mixed oil and water, and you're going to separate these two substances from each other, so you use a separatory funnel because the oil will float over the liquid water, and the water therefore will be separated first out from the mixture. Then follows the oil. Number 6 is the rubber bulb or the pipette bulb. This serves as a vacuum source in filling the pipette with reagent or a solution. Number 7 is the centrifuge. This is an apparatus used to separate the solid from the liquid using a centrifugal force. Number 8 is the desiccator. This is an apparatus where chemicals are to be placed, which are hygroscopic or will react with water from humidity. Now, this also prevents those chemicals which are hygroscopic to be moistened. Number 9. Number 9 is the Erlenmeyer flask. The Erlenmeyer flask is used as a receptacle for titration process, or this is also an apparatus used to measure large volume of liquids. Number 10. This is an analytical balance. This is used to measure the weight or the mass of substances, and its accuracy is up to the 10,000th place or the 4th decimal place. Number 11 is the volumetric flask, and this is used to measure exact or precise volume of liquids for dilution. Number 12, this is the dropper. The dropper is used to dispense small volume of liquids. Number 13, if you're going to notice in the picture that the arrow is pointed towards the iron ring. Now, this iron ring functions together with the iron stand, and this is used to support the vessel which is placed onto it. Number 14 is the thermometer. Now, the thermometer is used to measure temperatures. Number 15 is the crucible tongue. Now, the crucible tongue is used to hold the crucibles, especially when you are heating the crucible and you want it to be removed out from the flame. Then, of course, you have to hold it with a crucible tongue. Number 16 is the pH paper. Now, this pH paper is used to determine the pH of a substance, whether it is acidic or it is basic. We know that it is acidic when The pH ranges from 1 up to 6, and it is basic when the pH ranges from 8 to 14. When the pH is 7, we can say that the substance is neutral. Number 17. Number 17 is the iron stand. Now this iron stand is used to support the iron ring, the iron clamps, and the burette clamp. Especially when you are going to do titration process, then we have to use the iron clamp together with the iron stand. Number 18. This is a beaker. The beaker is used as a receptacle for liquids, and it is also used to contain liquids and measure large volume of liquids. Number 19 is the triple beam balance. Now this balance is used to determine the weight or the mass of an object or substances, and its accuracy is up to the hundredths place or the second decimal place. And the last one, number 20, is the top loading. This is an apparatus that is used to measure or determine the weight or the mass of an object, and its accuracy is up to the thousandths place or the third decimal place. So actually, we are having here 3 different apparatus used to measure mass or the weight of the substances or objects. First is the analytical balance, which is used to measure the mass of an object, and its accuracy is up to the 10,000ths place. Another one is the top loading, which is an apparatus used to measure the mass of an object, and its accuracy is up to the thousandths place. The last one is the triple beam balance, wherein its accuracy is up to the hundredths place. So those are the common apparatus which are to be used in analytical chemistry laboratory, because as we do the experiments, these are the common apparatus that you are going to ask from the counter to be used when you're going to analyze the substances. Before we are going to perform an activity or the experiment in the laboratory, let us try to review the rules and regulations on how to use the laboratory. I hope you have already a copy of the lab guide. Kindly open the lab guide and read together with me the rules and regulations to be followed when we work in the laboratory. So if you're going to look at there, if you're going to look at page 1, Roman numbers 1 to 3, they are very much similar to the rules and regulations as we had discussed in Chemistry 100B. So you may just review them and browse them back and try to recall what I have discussed in Chemistry 100B laboratory. If you're going to open to the next page, second page, look at letter C. There are rules in handling reagents and solutions. Okay, kindly get ready with the lab guide. We have number 1. Select the best grade of chemical available for analytical work. Whenever possible, pick the smallest bottle that is sufficient to do the job. Okay. Number 2, replace the top of every container immediately after withdrawing reagent. Do not rely on someone else to do it. So what does this mean? It means that you are going to put back the cover of the reagent bottle after withdrawing some amount of the reagents that you are going to use for the analysis. Number 3, hold the stoppers or reagent bottles between your fingers. Never set a stopper on a desk top. So that means when you're going to remove the cover of the reagent bottle, just hold it Do not place it on the desktop or the tabletop where the reagents are being dispensed. Why is this so? You are requested to just hold the cover to prevent any contamination of the cover from any chemicals which are dispensed on the dispensing table. Number 4. Unless specifically directed otherwise, never return any excess reagent to a bottle. The money saved for returning excesses is seldom worth the risk of contaminating the entire reagent inside the bottle. So do not ever return whatever excess reagent that you have withdrawn from the reagent bottle, because this can cause contamination of the reagent in the bottle. Contamination can lead to contamination can lead to a wrong analysis. Number 5. Unless directed otherwise, never insert spatulas, spoons, or knives into a bottle that contains a solid chemical. Instead, shake the cap bottle vigorously or tap it gently against a wooden table to break up the incrustation. Then pour out the desired quantity. These measures are occasionally ineffective, and in such cases, a clean porcelain spoon should be used. Okay, so you're asked to tap the bottle which contains the substances, and in those cases wherein there are incrustations of the solid, you kindly shake it a little bit so that it will be broken down to finer ones. Otherwise, you may use a spatula or a porcelain spoon to remove it out. Number 6. Keep the reagent shelf and the laboratory balance clean and neat. Clean up any spills immediately. So, if you're going to use the balance and you happen to spill on it, especially on the pan, kindly clean it immediately. So, those are the rules to be followed when you are going to handle reagents and solutions. Let's go to letter D. There are precautions in using an analytical balance. Again, we have already identified the analytical balance a while ago, that this is a very sensitive balance which could weigh or measure the mass of the substance up to the 10,000th place. Now, number 1, center the load on the pan as well as possible. So what do we mean by this? When we are going to weigh a substance on using the analytical balance, we are going to place the object or the substance at the center of the pan, very center part of the pan. Number 2, protect the balance from corrosion. Objects to be placed in the pan should be limited to non-reactive metals, non-reactive plastics, and vitreous or glass-like materials, because these substances can cause corrosion to the pan. Number 3, observe special precautions for weighing of liquids. That means we have to place properly the liquid into a container which is dry and clean to prevent spill on the pan of the analytical balance. Number 4, consult your instructor if the balance appears to need adjustment. Of course, we have to start that the balance must be at 0 mark. And if in case the balance does not give you a 0 mark or 0 reading at the start, we have to make adjustments. You can call me or the laboratory assistant in order to make the analytical balance start with a 0 reading. Number 5, repeat for number 5, correct. Keep the balance and its case scrupulously clean. A camel's hair brush is useful for removing spilled materials or dust. You cannot be denied that in performing weighing in the analytical balance, there are some substances which can be spilled, like powder or dust. So we have to immediately clean the pan. Because as I have said a while ago, that this analytical balance is very sensitive. Even the air that could enter into the analytical balance can be weighed. Number 6. Always allow an object that has been heated to return to room temperature before weighing it. So what does this mean? In cases wherein you happen to heat the liquid in a container and this is to be weighed in the analytical balance, what are you going to do? You have to let the temperature of this heated substance or container at the room temperature before weighing it. Please do not weigh hot objects using the analytical balance. This can give you a wrong result or wrong data. Number 7. Use tongs, finger pads, or paper strip to handle dried objects to prevent transferring moisture to them. When you're going to weigh substances in the analytical balance, of course, these are to be placed in certain containers. And when you are going to hold the containers to be weighed inside the analytical balance, you have to hold them using the tongue or paper strip, because this can prevent from the moisture from your fingers to be transferred into the container. Remember, even the moisture of your fingers transferred to the containers where your substances are placed to be weighed in the analytical balance can be detected by the analytical balance. So those are the precautions in using the analytical balance. Let's go to letter E. Directions for using a pipette. Number 1. Liquid is drawn into a pipette through the application of a slight vacuum. Never pipette by mouth because there is a risk of accidentally ingesting the liquid being pipetted. Instead, use a rubber suction bulb or one of the number of similar commercially available devices. So when you're going to pipette substances using the pipette, never pipette them with your mouth. You may accidentally ingest or swallow the liquid that is being pipetted. So use a pipette bulb or the rubber vacuum bulb in order to pipette the solution or the reagent through the pipette. Number 2, draw detergent solutions to a level of 2 to 3 centimeters above the calibration mark of the pipette. Now this is when you're going to clean a pipette, kindly draw out 2 to 3 centimeters above the calibration mark of the pipette so that you are sure that you can clean all the parts, the inside parts of the pipette. Number 3, drain the solution at this portion of the cleaning cycle if necessary. And number 4, fill the pipette with distilled water to perhaps 1/3 of its capacity and carefully rotate so that the entire interior surface is wetted. Repeat this rinsing step at least twice. Then let's go to the directions for using a burette. Number 1, thoroughly clean the tube of the burette with detergent and a long brush. Rinse thoroughly with tap water and then with distilled water. Inspect for water breaks and repeat the treatment if necessary. Number 2, carefully remove all old grease from a glass stopcock and its barrel with a paper towel and dry both parts completely. Number 3, lightly grease the stopcock, taking care to avoid the area adjacent to the hole. Number 4, insert the stopcock into the barrel and rotate it vigorously with slight inward pressure. And number 5, a proper amount of lubricant has been used. When the area of contact between stopcock and barrel appears nearly transparent, the seal is liquid tight and no grease has worked its way into the tip. So those are the directions in using the burette. Well, anyway, I hope that we can have a face-to-face in the laboratory so that you can have a hands-on in the use of these apparatus as you're going to do the analysis in the experiment. So those are all for today. Kindly read them again so that you can understand them clearly. And if you have questions, then you just write your comment down below. This is your teacher, Professor Nisitas Ruiz of Holy Name University.